Rahim. So let's begin with the part 2. Now you have seen this diagram before, but now I have added two more things. One is the cytosol and other is the cytogel. Actually, the cytoplasm is further divided into an interior portion towards the nuclear membrane, which is the cytosol. This portion is a little bit liquid portion, and this portion has all the dissolved substances, and it can move and it can provide different carbohydrates, lipids, and proteins to different processes that are acting inside the cell, whereas the exterior towards the cell membrane or the peripheral portion, it has cytogel, which actually has the role of storage, and this storage role is for the substances that are either entering the cell via endocytosis or leaving the cell via exocytosis. We will also study these processes in detail. So we have the jelly-like fluid portion, which is the cytoplasm, which is further divided into the cytosol and the cytogel, and we have certain neutral fat globules which are also present over there. Now let's discuss the organelles which are present inside the cell. The first organelle is the endoplasmic reticulum. The enzyme marker for this is the glucose 6 phosphatase enzyme. Always remember the glucose 6 phosphatase enzyme, which is the enzyme marker for endoplasmic reticulum. Now in this diagram we can see the endoplasmic reticulum. It is a flat vesicular structure and it consists of certain channels which are known as the cisternae. These cisternae, they are present over here, and there are two types of endoplasmic reticulum. One is the smooth one which lacks the ribosomes, and other one is the rough one which has a lot of ribosomes attached to it. And the rough part has a function in protein synthesis because the ribosomes are involved in the translation process of protein synthesis. And this smooth portion, it has a lot of functions that I'll tell you later. Now, as we can see over here is the distribution of these endoplasmic reticulum. Mainly these endoplasmic reticulum, this channel is continuous with the nuclear membrane, and it spreads around it, and it has the ribosomes which are also attached to it. And these endoplasmic reticulum molecules, they produce endoplasmic reticulum vesicles, and these vesicles are produced and they move towards the exterior, and their processing takes place in the Golgi apparatus. This is the main crux. Moving towards here, we have cisternae, the flat vesicular structures which are present over there, and it is continuous with the nuclear membrane. And the area of these endoplasmic reticulum is 30 to 40 times that of the cell membranous area. Also, the enclosing cavity inside them is known as the endoplasmic matrix, which has the endoplasmic watery medium, and this is the endoplasmic reticulum lumen. So also we have the rough or the granular portion which has ribosomes attached to it, and it is involved in the translation step, whereas the smooth agranular portion, it is actually involved in synthesis of the lipid substances. Now we move towards the functions of the endoplasmic reticulums. Main function of the endoplasmic reticulum is to produce the protein substances that are produced by the rough endoplasmic reticulum, and these substances that are produced, they move towards the Golgi apparatus and they are processed over there. Here we can again see that rough endoplasmic reticulum, because it has numerous ribosomes attached to it, so it is involved in protein synthesis, and the main function or main step of the protein synthesis is the translation step, in which the messenger RNA is converted into the protein molecules. And the smooth endoplasmic reticulum, it is now concerned with the lipid synthesis. The lipid synthesis is mainly the phospholipids, which constitute 25% of the cell membrane, and the cholesterol, which constitute the 15 or 13% of the cell membrane. These lipid constituents, these are packed inside the transport vesicles, and these vesicles then go towards the Golgi apparatus. Other function of the of the endoplasmic reticulum, they include the glycogen breakdown or the glycolysis, you can say, and they do the function of detoxifying the substances. Now, detoxification of the drugs and harmful substances is done by these steps. First is coagulation. They are combine all the toxic substances as they are combined together and they undergo coagulation, then they are oxidized, that is oxidation, then they do hydrolysis, and then conjugation with glycuronic acid, and in this way these substances are detoxified, whereas the other function is glycogen breakdown. Now we have the diseases related to the endoplasmic reticulum, that are the Alzheimer's disease, which is a neurodegenerative disorder. Then we have prion disease, which is accumulation of certain prion particles. Then there is cerebral ischemia, that is the loss of the oxygen to the brain, which is also a disease of the endoplasmic reticulum. And then we have a multiple sclerosis, which is the autoimmune disease. Only the names of these diseases are required at this stage. Moving towards our second organelle, which is the Golgi apparatus. The Golgi apparatus was discovered by Camillo Golgi. As we all know, it has a convex cis side and a concave trans area, and it has a the enzyme marker for the Golgi apparatus is galactosyl transferase enzyme. For endoplasmic reticulum, it was glucose 6-phosphatase enzyme. Now, the galactosyl transferase enzyme is the enzyme marker for Golgi apparatus. Now, the Golgi apparatus also consists of a stack of thin flat vesicles which are present in the Golgi apparatus. And the main function of the Golgi apparatus is to process the secretions or the endoplasmic reticulum vesicles that are coming towards it, and through its cis side it takes the vesicles, and through its trans side it releases the vesicles, and the trans side vesicles, they contain substances that have undergone changes. So the main function of Golgi apparatus that we have studied until here is the processing of substances that are coming to it via the endoplasmic reticulum vesicles, but sometimes it can also produce certain carbohydrates or synthesize certain carbohydrate substances, which are the hyaluronic acid and chondroitin sulfate. These hyaluronic acid and chondroitin sulfate are major constituents of the proteoglycans that are actually glandular secretions. They are part of ground substance of matrices, and they are the part of cartilage and bone, as well as they are important for migration and proliferation. So they are part of ground substance, the proteoglycans, the cartilage and bone, and they are required for migration and proliferation, etc. So Golgi apparatus has the processing function and the synthesis function, and synthesis function is related to carbohydrate synthesis. So let's understand this particular diagram to understand the function of Golgi apparatus. So now these are the endoplasmic reticulum. In these endoplasmic reticulum, the agranular portion, we have the secretion or the production of protein. After the protein formation, we have lipid formation which occurs in the smooth endoplasmic reticulum. These protein and lipid portion, they are packed in the transport vesicles, which are the endoplasmic reticulum vesicles, and they go to the cis area of the Golgi apparatus. From the cis area of the Golgi apparatus, they undergo processes of compaction and processing, and they are converted into complex or modified forms, and they are secreted from the trans phase of the Golgi apparatus, and they become the secretory vesicles. These secretory vesicles then, by exocytosis, they leave the cell and they go to the area that are where they are required. These secretory vesicles, okay, so now they are saying that whenever they did an experiment, and they found in an experiment, if they put amino acids, the amino acids were detected in the endoplasmic reticulum after 3 to 5 minutes, in the Golgi apparatus after 20 minutes, and they were excreted out of the cell within 1 to 2 hours. So this was the functioning of Golgi apparatus. Another function of the Golgi apparatus is to secrete a very important substance, that is a single membrane bound organelle, which is the lysosome, which contains the digestive vesicle. These secretory vesicles, they can contain modified proteins or modified lipids, or they can contain hydrolytic enzyme containing substances, and then they will be known as the lysosomes. And they are saying that these secretory vesicles, they undergo the process of exocytosis, and the main ion which is responsible for regulating the process of exocytosis is the calcium ion, and it provides them with energy for the opening of the membrane to the outer surface and extrusion of the contents from the inner surface of the cell to the outer surface. Whereas certain vesicles, they can be utilized within the cell, but most of the substances, they are extruded or excreted out of the cell via with the help of the calcium ions via the process of exocytosis. Also, Golgi apparatus has an important function to replenish the cell membranes. The main thing that happens in endocytosis process, that is the taking in of food, is that certain cell membrane is reformed. But in exocytosis process, the cell membrane, it is lost because this cell membrane is involved in the packaging of the substances. So when this cell membrane is lost, the new cell membrane is actually provided by these secretory vesicles, which can fuse with the cell membrane and help in the replenishing of this outer boundary of the cell, which is very much important and without which the cell would die. So now in the diseases related to Golgi apparatus, we have the Alzheimer's disease, the Parkinson disease, and the Huntington disease. These all diseases are neurodegenerative diseases, and they occur due to the dysfunctioning of Golgi apparatus. The multiple system atrophy is very much important because we know the Golgi apparatus is involved in the synthesis or the processing of synthesis of the carbohydrates and the processing of the proteins and the lipids. So if there is any defect in the synthesis of these or synthesis or the processing of these substances, we will have the atrophy or loss in functioning of multiple systems. This is how the Golgi apparatus is very important. Now, coming towards the organelle, which is actually produced by the Golgi apparatus in the form of a secretory vesicle, and it contains the digestive enzymes. It is the lysozyme, a single membrane bounded organelle which is present inside the cell. For the enzyme detection, we have enzyme marker, the acid phosphatase enzyme. Now, the main function of the lysosome is to destroy the damaged cellular structures, for the digestion of the food particles, and for the elimination of the unwanted particles. The dimensions are 250 nanometers to 750 nanometers, and it contains 40 different digestive or hydrolytic enzymes which are produced. Now the proteins, they are converted into amino acids. They undergo hydrolysis. The glycogen, it is hydrolyzed into its simpler form, glucose. Lipids, they are hydrolyzed into simple form, that are fatty acids and glycerols. Now the digestive enzymes of the lysosome, they are very much active enzyme, but they are prevented from proliferating or destroying the whole of the cell by the single membrane of the lysosome. So, how does the lysosome hydrolytic enzyme perform their function? That is by the process of fusion. So, now we have three main functions of the lysosomes, which are the intracellular digestion, autophagy, and autolysis. So, now what is intracellular digestion? Digestion of the lipids, proteins, and fats, and carbohydrates. Now we have this portion, that is the autolysis. Autolysis is the degeneration or the regression that is done by the lysosomes to remove certain organelles which are no longer in the use. For example, this regression occurs in after pregnancy, in some muscles during long periods of inactivity, and in the mammary glands at the end of lactation. So in all the substances when we need to reduce the amount of the substance in the body, we have the process of autolysis to do it. So what happens is the lysosomes, they release their hydrolytic enzymes, and these hydrolytic enzymes, they then attach to the substances, they do the intracellular digestion, and the substances, those particles are eliminated. Lysosomes also have the bactericidal agents, which are lysozyme and lysoferrin, and because they contain acidic substances and hydrolytic enzymes are also acidic, so they provide a proper pH which is 5. So what is lysozyme? It actually destroys the bacterial cell wall. Lysoferrin, it is related to the iron, so it actually binds the iron to itself so that the bacteria cannot use the iron and do their synthetic reactions. And a pH of 5, which activates hydrolysis enzyme and inactivates the metabolic system of bacteria. Secondly, now we have the process of autophagy. Autophagy means to eat oneself. Where there are many organelles which are not to be used, they are or they are to be discontinued, like the mitochondria or other substances. These are actually digested by the process of autophagy. Auto means itself, and phagy means to eat. So the autophagy is the process of eating the cell organelles. So lysosomal membranes, they fuse with them, and then they form a secondary lysosome, and then they destroy those organelles. It is very much important because it provides the cell with the nutrients, and it is important for homeostasis, that is maintenance of constant internal environment, and also for the maintenance of the new organelles. For example, if there are old organelles present and new organelles are also present, but the old cannot work, so they will take up the space only, and they will take up the nutrients only, and their work effectivity will be less. So to maintain the life form, we have the process of autophagy. So now we can clearly see that the substances which are to be digested, they are converted into an autophagosome. Now this autophagosome, they are surrounded by a membrane, then they're converted into this autophagosome. The lysosome fuses with this autophagosome and forms a secondary lysosome. It releases its lysosomal hydrolytic enzymes. They digest those substances, those uh, food particles, rudimentary organelles, and as a result of which we get rid of those organelles and we get the nutrients that they were taking. This is how the vesicle breakdown and degradation of autophagy and autolysis takes place. Very important point for MCQ is that mitochondria has a lifespan of 10 days before it is destroyed by the process of autophagy. Now discussing the lysosomal diseases, which are the lysosomal storage diseases mainly. One is the Tay-Sachs disease. It is a congenital abnormality in which the lipid synthesis, in which the lipid breakdown is impaired, and the lipids, they accumulate, and they sometimes accumulate to brain, and the brain development is impaired. Now we have a Gaucher's disease, in which there is undigested glucose cerebroside, and because of this undigested substance, we also have certain brain abnormalities. And then we have the mucopolysaccharidosis diseases. These are the diseases related to the lysosomes, and first and last are the lysosomal storage diseases. Now we have peroxisomes. Peroxisomes, the enzyme markers for this organelle is catalase or uric acid oxidase. It is a self-replicating enzyme, self-replicating organelle, and it is also formed by the Golgi apparatus. It is also single membrane bounded, but the enzyme that it contains are oxidases rather than the hydrolytic enzyme that were contained in the Golgi apparatus. The main thing that they contain is catalase and oxidase enzyme. The oxidase enzyme, it actually converts the oxygen hydrogen into H2O2, which is the hydrogen peroxide. The peroxidase enzyme, it actually makes this toxic compound, the hydrogen peroxide, which is converted into normal non-toxic compounds, catalase, by the enzyme catalase, which converts this into water and oxygen, which are non-poisonous. It is involved in the detoxification of acetaldehyde by the peroxisomes of liver. Also, major function of peroxisome is to catabolize long fatty acid chains. Long fatty acid chains are broken into small fatty acid chains by the peroxisomes. So now we have the diseases that are related to the peroxisome, which are mainly comprise the peroxisome biodegenerative disorder. And other is the Zellweger syndrome. This Zellweger syndrome is characterized by the absence of the peroxisomes, and now the oxygen and hydrogen ions, they are not converted into H2O2 and then oxygen and water. And in this way, the oxygen and hydrogen, they are converted into reactive oxygen species, which are very harmful to the body. Now we have the secretory vesicles. They can be secreted by the endoplasmic reticulum. They can be secreted by the Golgi apparatus. They can be secreted by any substance, but mainly Golgi apparatus and endoplasmic reticulum. Now these secretory vesicles or secretory granules, they contain proenzymes. Proenzymes are the enzymes which are not in their active form. So these proenzymes are released by the secretory vesicles or secretory granules into the cytoplasm, and when they are required, they fuse with the substances and they perform their function. Mainly in the pancreatic SNR cells, we have the proenzyme secretory vesicles, and these proenzyme vesicles, they are released through the outer membrane of the pancreatic duct into the duodenum, where they become into their active form and perform the digestive functions in the intestinal tract. From the pancreas into the duodenum, and in the duodenum they are released and they perform their function of digestion.